Hello, everyone. Welcome back to the UCIVO High School Club meeting. Today, we will be covering Campbell's chapters 21 through 24. Just a quick reminder, please sign up for the UCIVO competition. I believe we have a link for you. Ivan, could you send a link? Right now, the sign up is open, and yeah, please sign up for the competition. Okay, let's get started. Today we'll be covering Campbell's chapters 21 through 24, starting with genomes and their evolution. So in the 1990s, okay, what else do I need to say, Ivan? Go ahead. You did? Oh. When you register for UCIVO, you won't get a reply email saying that you did sorry, that you did register, but just register the first time, and then we will confirm that you registered for you. There's something weird going on. Okay. Ellen, we'll confirm that with you guys. Okay, let's not go there. All right, let's actually get started. So in the 90s, a bunch of researchers came together and they decided, hey, we're gonna sequence the entire human genome. This took 16 years and labs all across the world. One important sequencing technique that they used was called whole genome shotgun sequencing, which is a really cool name, but basically this just means that they take a genome, they fragment it into a bunch of little pieces, and they sequence them using the various techniques discussed last time, probably dideoxy if I had to guess. And they sequence them, as you can see here, they match up the ends this is all done by computer algorithms. They match up the ends and then form the entire sequence. Identifying protein coding genes. So sometimes you have a piece of DNA, you want to know what it does. So the first thing that you do is you need to find out whether or not this section of DNA codes for a protein. Use a method called gene annotation. Basically, you look for patterns that hint that the gene does make a functional protein. For example, you look for start codons, stop codons. Any transcribable DNA will need one of these. You can also look for splicing sites. You can expressed sequence tags are like DNA domains commonly found in like, oh my God, how June is Derek. Oh, I didn't realize. Anyways, express sequence tags, they're DNA domains commonly found. There's a huge like website online with like catalogs of every single discovered protein, and the DNA sequences for those proteins and their domains are like listed in a database, and these express sequence tags correspond to a common protein domain. A protein domain is just like a part of a protein. Like if you think of you as a protein, your arm would be a domain, right? It's one section that does a specific function. After you look for these specific DNA things, you compare them with your DNA segment, and then you can use RNA-seq, which is basically you take RNA, transcribe it back into DNA to find out whether or not like the gene is actually being expressed. Genomes vary in size, number of genes, and gene density. As a general rule, eukaryotes have a lot more genes than prokaryotes. but gene density of prokaryotes notably is greater than eukaryotes. The gene density is the number of genes per like total base pairs, and eukaryotic genomes have a lot of non-transcribable regions. I have a diagram on the next slide showing all of the types of DNA stuff in eukaryotes, but prokaryotes don't have introns. They don't have a lot of the regulatory sequences that eukaryotes have. If you look at this table on the right, notably you'll see that like this plants have a lot of, they have very big genomes, mostly because they can they can survive polyploidy. They can duplicate their DNA and survive like that. Non-coding DNA. So this is a chart I was talking about. So protein coding genes, or exons specifically, they're only 1.5% of the eukaryotic, or this is a human genome in particular. All the other 98.5% does not code for a protein. So this just comes to show like how low eukaryotic gene density can be. Some pieces of non-coding DNA are regulatory sequences, so start codons, stop codons, tata boxes, like pieces of DNA that have specific functions in regulating transcription. And introns, this is part of the transcript that isn't translated. Gene fragments and pseudogenes. Gene fragments, sometimes pieces of DNA can break off. Pseudogenes they're generally duplications of a gene. Oh, okay. Pseudogenes are generally duplications of a gene where when polymerase is copying a specific gene, sometimes it falls off and it starts at the beginning of that entire gene and makes a second copy. And over time, some of these genes can lose their original functions. Repetitive DNA, we'll get into that in a bit. Okay, eukaryotes have a lot of non-coding DNA. So one notable non-coding DNA is transposable elements. So these are like bits of DNA that can move around within like a genome, within a nucleus. They can like jump between chromosomes. And there are two types, transposons and retrotransposons. These two differ in how they move between locations. Transposons move with a DNA intermediate. So this is a transposon. It like copies the DNA. The DNA moves to a new location. Retrotransposon is the transposon is transcribed into RNA, and then the RNA goes to a new location where it codes for a new DNA. These two just differ in their mode of transport. Next, ALU elements. They're transcribed into RNA but don't code for proteins. Yeah. Short tandem repeats. These are one important thing about short tandem repeats is they're used in criminal, never mind. I forgot what it's called. I'm blanking. Duplication, rearrangement, and mutation of DNA contribute to genetic diversity. So forensics, sorry. Thank you, Derek. Short tandem repeats are important in forensics where different people have different numbers of short tandem repeats. And you can use those numbers to determine if DNA belongs to one person or another. Duplication of entire chromosome sets, alterations of chromosome structure, exon shuffling, and exon duplication. So this is how, these are some ways that new functions of proteins, and even new species can arise. So duplications of entire chromosome sets, usually this kind of aneuploidy is like is deadly. But in some cases, as we'll see later, this can be helpful. Alterations of chromosome structure. I think here on the right, you can see how a human chromosome number 2 is a fusion of chimpanzee chromosomes 12 and 13, exon shuffling and exon duplication. So this is just, if you have exons code for a specific protein domain, and if you have, let's say you have one specific gene that codes for a kinase, and you have a certain number of the active site domains. Well, if you like duplicate one of those domains, then you have a kinase that's hyperactive. And then basically the changes in the exons within the transcribed exons within a gene can change the function of a protein. Comparing DNA sequences, comparing genomes, more similar DNA means that two species are more closely related. This makes sense. If a species diverges recently, before they diverge, their genomes are mostly identical, and then after the time that they've diverged is proportional to the time that mutations can accumulate. Distantly related, compare similar genes to clarify evolutionary relationships. Closely related species have few differences in genes. There is also variation within a species. SNPs, single nucleotide polymorphisms, is where within a population, at least 1% of that population has a nucleotide base mutation at a specific point. This is important in diagnosing diseases or risk factors. I think I'm not sure if we went over this before, but basically this SNPs are generally inherited together with a disease-causing allele. So by finding a SNP, you can try to discover it's associated with the disease, basically. And short tandem repeats, we went over this earlier. There are regions of our DNA where it's just a bunch of repeating, like ATG, like a short tandem repeats. I don't really know what to say. And copy number variants, there are short tandem repeats and copy number variants result from, as we talked about before, DNA polymerase sometimes when it runs, it'll just fall off and it forgets where it doesn't remember where it fell off. So it'll start from the beginning and make a copy of what it was just making. And that results in different numbers of short tandem repeats and different numbers of and results in copy number variants. Anyways, on to chapter 22, descent with modification. We have 4 chapters today, so I'm moving a bit fast. I'm trying to, at least. So if you have any questions, please shoot me in chat. Descent with modification, Darwinian view of life. Before Darwin, many people thought that all animals were organized into a ladder, and at one end of the ladder was humans, and at the other end of the ladder, like every organism occupied one rung on like a giant ladder, and they believed that was how, I think, creationists, that was how God created them. Linnaeus, notably, he came up with our current drunk king Philip, that domain kingdom phylum thing, binomial nomenclature, which is you name a species, the scientific name of a species is their genus followed by their species. Related resemblance among species, the pattern of creation. Darwin disagreed with this natural ladder, and he believed that classification was based on something he called evolution. Um, here's another person who theorized before Darwin. Lamarck, he believed that acquired traits are passed down to your offspring, right? Um, basically, as a giraffe stretches its neck throughout its life, its neck grows longer, and because it stretched its neck, its offspring will also have a longer neck. Use and disuse, blah blah blah. Sorry, I got poked to go faster. Darwin's focus on adaptation, inherited characteristics, enhanced survival and reproduction, related to formation of new species. So Darwin believes that specific traits help organisms survive and reproduce, and that we'll get into this in a second. Darwin focused on adaptation. More animals are born than can survive. So the most fit animals, the animals most adapted to their environment by the principle of natural selection will have the most offspring, and over generations, their traits will be more common. Descent with modification. He noticed that animals had the same generations. Offspring had similar characteristics to their parents, so there has to be a way that parents are giving their traits to their offspring. Yeah, artificial selection. Yeah, artificial selection, natural selection, and adaptation. Artificial selection is what us humans do. Like, for example, we can take a wild mustard plant and breed it with other mustard plants that have favorable characteristics. Like, for example, if we look for mustard plants with big apical buds, and then over many generations, we breed the mustard plants with the largest apical buds with each other, and it becomes a cabbage. Natural selection is kind of like artificial select, not really. Natural selection is basically survival. The ability of an animal to survive and reproduce will determine the relative frequency of that animal's traits and genes within a population. Adaptation. A species as a whole can adapt to their surroundings. Yeah. Key features of natural selection. Individuals with certain heritable characteristics survive and reproduce at a higher rate. If environment changes over time, natural selection gives rise to new species. Yeah, I think this is pretty straightforward. Individuals not evolve, populations evolve over time. So the definition of evolution is a change in allele frequencies. So the allele frequencies for an individual will remain the same throughout their entire life. There's nothing that can change that. But if you're talking about a population, as time passes, as unfit traits are selected against, allele frequencies can change. 20. Here's one evidence of Darwin's theory of evolution, which is homology, similarity resulting from common ancestry. So if you look to the right, there's a diagram of different forelimbs in very different animals: cetacean, cat, human, cetacean, I believe, and a bat. But all four of these animals, although they are morphologically very different, at a very basic level, you look at their skeletal structure, you can see very distinct common features. One important thing to note about homology is, do we have analogy on the next slide? No, we don't. So one important thing is to distinguish homology from analogy. Homology versus analogy. Homology is similar traits resulting from, because those traits were inherited from a common ancestor. However, analogy is two species evolve into similar forms because they have similar environments. Basically, analogy is two species become similar, even though they're not closely related. A common way to tell homology versus analogy is that if the characteristics of the shared trait are very complex, it's most likely a homology. Whereas if it's a very simple association, it could be an analogy. Yeah, anatomical and molecular homologies, vestigial structures. Vestigial structures are like our tailbone is a vestigial structure. The hip bones of a whale are vestigial structures. These are bits of our evolutionary past that we know that we have because our ancestors had, but we have no use for them. So over time, they lose their function and they shrink. Anatomical and molecular homologies. Do we have or thought? Was this on this? There are two types of in DNA. There are certain molecular homologies. So you can have, notably, I think, I think it was paralogous. Correct me if I'm wrong, but paralogous genes are two similar genes that. Let me type that. Paralogous. The appendix is a vestigial structure. So the appendix that's an interesting question. A lot of animals that eat like vegetation have what's called a cecum. We also have a cecum, but it's much smaller because the cecum is used it houses bacteria that can digest cellulose. And then basically the plant matter is put into the cecum to ferment, and then the bacteria are eaten by the this is too common. Basically, the cecum is used by herbivores to ferment their food. We are not herbivores, therefore we do not need a cecum, and the appendix is likely what is left of that. Anatomical, yeah. Sure. Convergent evolution. Oh, this is what I was talking about before. Convergent evolution gives rise to analogous structures. So when similar evolutionary pressures cause two distantly related species to evolve into a similar morphology, a similar morphology, that is a convergent evolution, and the similarities between those species is called an analogy. The fossil record, extinction of species, origin of new groups. This is a good record, a good way to record how life has changed. It provides us intermediates between, for example, hippopotamuses and these even-toed ungulates are closely related to whales. But if you look at living animals, it's hard to see how they're related. But if you look at the fossil record, you'll find examples of intermediates between these animals that help illustrate their evolutionary ties. Here's more evidence that supports evolution. Biogeography, endemic species. I think we all know that Earth used to be like all the continents on Earth used to be in one mass called Pangea. As those continents drifted apart, the species that used to be able to move between continents are separated by natural barriers, and this results in This results in very different populations in different continents. The further away these continents drifted, the more different the life there is. Chapter 23, the evolution of populations. Okay, I'm on track. Genetic variation makes evolution possible. So evolution is defined as a change in allele frequencies within a population. But to have allele frequency, it necessitates that you have different alleles, or else the frequency, the only frequency there would be is 100% for one allele. Sources of genetic variation, formation of new alleles, altering gene number and position, rapid and sexual reproduction. So all variation comes originally from mutation. Most mutations are neutral. Some are detrimental, but a small few can be beneficial. And these are the beneficial ones that are the ones that are propagated in new generations, altering gene number and position. So in this case, like I talked about many times before, when polymerase falls off, you can duplicate genes, and maybe having 2 copies of one gene helps you make a function, helps you accomplish a function better. Or maybe having 2 copies helps will waste too many resources, and therefore you are at a disadvantage. Rapid and sexual reproduction. Rapid reproduction just increases the amount of mutations that happen in a population, because although there's no recombination of DNA, that you're replicating so much DNA so fast that even if your mutation rate is low, which it usually isn't, a lot of mutations will propagate. And sexual reproduction will jumble up sexual reproduction will mix up your alleles, and no 2 gametes are alike. This is the Hardy-Weinberg equation. I think we went over this before. Let me know, send me a chat if we haven't. I believe we have. Okay. I'll just go over it again. This Hardy-Weinberg models a non-evolving population with no selective pressures. There's more conditions than that, but simply put, in a population with you're modeling a specific gene, it's a completely dominant gene where little p is the frequency of the little p is the frequency of the dominant allele. Little q is the frequency of the recessive allele. So p squared is the frequency of the of homozygous dominant in a population, and q squared is homozygous recessive in a population. 2pq is heterozygote frequency in a population. All of that should add up to 1, because a organism can only be one of these 3 variants. Natural selection, genetic drift, and gene flow. Adaptive evolution is just natural selection. There's a trait that's favored by selection. Because you have this trait, it helps you in some way, and therefore it helps you better survive and reproduce in your environment. That trait is going to be selected for. That's adaptive evolution. Genetic drift is just random. Adaptive evolution is the most influential motivator, most predictable motivator of evolution. Genetic drift, on the other hand, and gene flow are both random. They're very unpredictable. Genetic drift is just a random, it's evolution. Remember, evolution is the change in allele frequencies. So genetic drift, it is a change in allele frequency, and therefore it is evolution, but it's not motivated by any, like, there's no reason. It's just by chance, this one allele happened to be passed on more often than this other allele. This often happens when there is a small population. So if a common example is if a group of birds from flew to the Galapagos Islands, the population there is small, and therefore the chance of something random happening is much larger and has a much larger effect. The bottleneck effect, on the other hand, is if you have a major disaster killing off a significant portion of the population, and the genotypes of the animals that died are lost to the population. So that bottleneck could rapidly change the genetic makeup of that population. Gene flow is, okay, maybe I should have defined population. A population is a group, a species, a group of animals that are within one species that can readily interbreed with each other, right? But sometimes these animals will emigrate from one population to the other, and when it does, it takes its genes from one pool and puts it in another. That's called gene flow. Does that make sense? Okay. Natural selection. Here is 3 types of selection. Stabilizing selection this is the average phenotype, the middle phenotype is most strongly selected. This is generally in stable environments. Directional selection, one extreme is favored. Excuse me. Directional selection is where one extreme phenotype is favored. This is common in changing environments. Disruptive selection is when the average phenotype is selected against, but both of the extremes are favored. This is generally in patchy environments, meaning that within one biome or one ecosystem, there's two different surroundings that an animal can live in. Yeah. Sexual selection, sexual dimorphism. I think sexual dimorphism is when the male and a female of one species will look different. So male deer will grow antlers, right? But females won't. Male peacocks have a colorful display, females don't. And then there's intra versus intersexual selection. So intrasexual selection is generally when the males will fight. A male will fight another male for the right to mate with females. I think generally, walruses, I think, moose, a lot of animals practice intrasexual, but intersexual is also common. This is when a female chooses a male by like certain characteristics. Like female peacocks will look for males with colorful plumes. And interestingly enough, this is because that this a specific phenotype is often associated with fitness, and it can be like completely unrelated. So, for example, if you have there's an example in Campbell's where a bullfrog with the favorable mating call would produce a lot more viable offspring than a bullfrog with an unfavorable mating call. And it's weird because these two things, you wouldn't think they're related, but intra is males fighting Basically, yes. I lost my train of thought. I don't think that was that important. I don't think what I was talking about was that important. So, balancing selection, frequency dependent selection, and the heterozygote advantage. Um, the heterozygote advantage is when you have. I think I can explain this better with an example. Common one that a lot of people talk about is sickle cell disease. So when if you have two copies of a sickle cell allele, you get sickle cell disease, which your blood cells will aggregate very easily and it'll cause internal bleeding, it'll clog up your capillaries. It's very heavily selected against. However, the heterozygote will give you a the heterozygote will give you uh, a resistance to malaria, which is a devastating disease in Africa. So in places where malaria is common, although the homozygote is selected against, the heterozygote is selected for. So this interplay between the two, the favored and unfavored, will balance out. And I think the percentage of sickle cell alleles in populations is roughly like 10%. Yeah. And frequency dependent selection. Oh, this one's really cool. So this is basically the whatchamacallit. The fitness of an allele is dependent on how common it is in the population. So Campbell says this really cool example of there's a species of fish that there's a predatory fish and a whatchamacallit. There's a predatory fish and a prey fish. And the oh my god, I am blanking. I'll get back to this, but I'll try to see if I can remember the example. But basically, it's frequency dependent selection is just the fitness of a phenotype depends on how common it is. So if a phenotype is very common in a population, it could be selected against. But if the frequency of that same trait is not common in a population, then it could be selected for. If we have time, I can show you the example later. The fish. The origin of species. I think we'll have time. Biological species concept emphasizes reproductive isolation. What is the biological species concept? A biological species is defined by a group of animals that can interbreed with each other. And this is not a perfect definition. We'll see there are many exceptions to this rule. There are many like iffy things, but it's not a bad definition by any means. Excuse me. Speciation, microevolution, and macroevolution. Speciation is the divergence of one species into two separate species. Microevolution is changing allele frequencies within a species, and macroevolution is basically just speciation. Reproductive isolation, classified. So there are many reasons why two animals can't mate with each other, right? You can't mate a frog to a deer for many reasons. And a lot of the reasons like you might think are common sense, but it's actually quite generalizable. There's a lot of, I think, do we have this one? I think we might have one later, but reproductive isolation, there are many factors that prevent reproduction between two species. They're classified by whether factors act before or after fertilization. So prezygotic barriers prevent like uh, fertilization from taking place. And postzygotic barriers are after the zygote is formed, after the egg is fertilized, there is something something to do with something happens next that decreases the survivability of the offspring. Surprised we don't have something. But prezygotic barriers include habitat. Deer and frogs don't live in the same place, therefore they can't even meet, right? Temporal, maybe deer and frogs aren't the best choice, but I guess it is. Deer and frogs both have mating seasons. If those don't match up, that dude, I have 15 minutes. Go away there. I guess a better example of a temporal barrier would be species that are nocturnal versus diurnal, so active at night or in the day. They won't meet each other, and they won't have a chance to mate. Behavioral, so maybe some species have a distinct mating call, or they have a specific dance that doesn't match with another species. Mechanical, use your imagination. Genetic, sometimes when fertilization happens, if the alleles don't match up properly, if there's like a big mismatch, then things don't connect properly, and the zygote fails. Well, it's not even a zygote. However, hybrids are like the big exception to the biological species, where two groups of animals considered species can interbreed because they're like kind of related close enough that they can produce some offspring, but often those offspring are non-viable. They're usually weaker. Maybe the hybrids break down after a few generations, or they're sterile. Either way, they can't reproduce as successfully as either of the parents. Limitations of biological species concept: absence of gene flow. And here are the other examples of species. So morphological species is defining a species by its morphology. So how it looks, basically how it looks. What is its body plan? A limitation of this is if you have analogy, and two species converge, they have very similar morphologies. They can be, if you use a morphological species definition, they'll be considered the same species, even though they are distantly related. Another thing is, I saw a video where the different breeds of dogs are all dogs are one species because they can interbreed. But different breeds of dog will have different skeletons. If those in the future, if those skeletons fossilize, if those skeletons fossilize, people will believe that because the fossils look very different, like if you have a Pomeranian versus a German Shepherd, they'll believe that these two fossils are two different species, which is obviously not true. The ecological species concept, you define a species by the ecological niche it takes up. Basically, the role of a species within an environment determines its species. And this is also like an how do you define niche? There's too much data that you need to know. And the phylogenetic species concept is you just classify a species by its relation to other species, to other animals, like using molecular fossil morphologies. Yeah. Speciation can take place. Here are the two broad categories of speciation. If you have a population which diverges into two species, it can either be allopatric or sympatric. As you see here, allopatric. I'm not sure if you can see my mouse, but here now you see. Allopatric is where two species diverge because they are geographically separated. So in this case, this little divot here separates the two species, and the fish become different colors. Sympatric speciation, on the other hand, the two species are still living in the same habitat, but by other means, maybe temporal, behavioral, they've managed to diverge. Speciation can take place with or without geographic separation. So these are some examples of sympatric speciation. So polyploidy is very common in plants. Not much in animals, because in animals polyploidy is fatal. But in plants, if you duplicate the genome, the plant becomes a new species. And there are two ways that this can happen. One is an autopolyploid. So when a plant asexually reproduces, or it mates with another member of its own species, and some error happens in meiosis that leads to a zygote with double the alleles, double the chromosomes, sorry. This is auto. So your own species, one species gave rise to a new species by duplicating its genome. However, allopolyploid, on the other hand, is when two different species will mate and form a hybrid. And that hybrid is generally infertile. It cannot, in its early stages, it cannot sexually reproduce, because if it tries to mate with either of the parents, the chromosome numbers don't match up with them. So let's say you had a plant with 45 chromosomes, another with 47, right? And let's say, actually, no, never mind. Let me restart it. So allopolyploid is when a plant hybrid forms, and because it's a hybrid, it cannot mate successfully with either of the parents. But if it reproduces by asexual reproduction, like propagation, then it can give rise to its own species and breed with its own species, because they have the hybrid and its offspring have the same number of chromosomes. And sometimes maybe there's a duplication error, and that leads to polyploidy. Habitat differentiation, sympatric speciation results from new ecological niches. So, for example, if you have two species living in a rainforest, if one lives in the canopy, one lives on the forest floor, they occupy different niches. They have different roles in their environment, and those roles mean they don't interact. I guess that wasn't, but sympatric speciation results from new ecological niches. And sexual selection, in a population, if you can have like we talk about intra or intersexual, I think this is usually intersexual selection. The females will select different kinds of males, and the females and the males that they select will gradually diverge, because the offspring of those males will attract like the females that are attracted to their behavior. Just know that different sexual selection within one species can split a population. Okay, I gotta hurry up. Hybrid zones reveal factors that cause reproductive isolation. Hybrid zone, region in which members of different species mate and produce hybrids. Mating between species with incomplete reproductive barriers. Hybrid zones over time can undergo one of three things: reinforcement, fusion, or stability. So a hybrid zone is exactly what it sounds like. If one species occupies one area, another species occupies a different one, but they're close enough that species A can go and mate with species B in like the middle area between their two environments. And that place where the two species meet and mate is the hybrid zone. This is like pushing the definition of biological species, because like, yes, these two species can interbreed with each other, but they often don't produce viable offspring. So generally they're counted as two different species. Over time, these hybrid zones can either reinforce themselves, which means that the hybrids will be so unviable that it splits the population further apart. Basically, there can be no gene flow between the two populations, and as a result, over time the populations just naturally drift apart. Another possibility is fusion. So maybe the two species aren't that different after all. They mate, and the hybrids can survive and reproduce, and those hybrids serve as vectors that like spread the gene pool of the two species, ultimately fusing the two different species into one. And stability, sometimes maybe the reinforcement doesn't happen, so the two species don't drift further apart. The two species don't fuse into one. Rather, they maintain the same amount of like difference, but they keep the hybrid zone and the hybrid zone kind of just stays there. It stabilizes. 24, speciation can occur rapidly or slowly and can result from changes in few or many genes. Patterns in the fossil record. How do we date fossils? You might think of, right off the top of your head, like this is what I thought of. I thought of carbon dating, but it turns out that a lot of like the fossils themselves, if you try to go back really far, like millions of years, I'm thinking hundreds of millions of years, it's hard to use carbon as your radioactive dating isotope, because it has too short of a half-life. But in order to fix that, you use the layers of rocks between, like above and below a fossil, to determine the age of that region. This is called the punctuated equilibrium model, where you infer the age of a fossil by estimating the ages of the rocks above and below it. Right. So there are longer lived isotopes that can exist in rocks, but they don't exist in living animals. And so they'll be deposited in the soil layers, and that allows you to determine the age. Speciation rates, punctuated pattern and lab data, rapid speciation, studying the genetics of speciation. Speciation might require the change of only a single allele or many alleles. Punctuated pattern and lab data, rapid speciation. I guess you can determine how fast species change by looking at the ages of different fossils and seeing like morphological trends between one layer to the next. Look at random, because we're at the end. Are eyebrows sexually selected traits? According to Derek, yes, but I honestly I'm not sure. I don't see why. Maybe? Yeah, Derek, I would have said maybe, but um, from speciation to macroevolution, cumulative effect of many speciation and extinction events. Uh, this is what our world looks like. What our living biosphere is the result of the evolution of new species and the extinction of ones that couldn't survive. We're out of time. Not we're out of time, but we finished. Um, one thing I'm just gonna add this for fun at the end. Uh, actually, never.